finishing up with diversity, we're going to go over finish up with the seeded vascular plants with angiosperms. So angiosperms are the last group. These are flowering plants. They're also known as anthophyta, which literally means flower producer or flower plant. The newest features, of course, these have all the features that other land plants have, and the same thing that other seeded plants also have. But some new features are flowers, and flowers are specialized structures for pollination. They're mainly to attract pollinators, even though there's still a fair number of plants with flowers that are wind pollinated. Others are water pollinated or animal pollinated. And this is like a cone. It is a modified branch with leaves. And these leaves occur in whorls, which would basically be kind of similar to leaves coming off of a branch. And there are 4 possible whorls. Starting on the bottom or on the outside, there are sepals. These are usually green and they're covering the bud. So like if you've ever gotten a rose, the green part is a sepal. Petals are generally colored, although they can be white and they can be green, they can be black, they can be lots of different colors. And then the next thing in is stamens, which are also known as the microsporophyll. This is the male structure. And then there are carpels. You also need to know the other flower parts. So I'll just show you. You have this handout here of a flower. It also has another one on this side. The reason why I gave you 2 was because this one is a little easier to tell the sepals and petals apart. But this one also is kind of unusual in that it has the ovary embedded in what they call the receptacle. So the pedicel is the part that holds the flower. The receptacle is kind of a fat area that ends up holding the other flower parts. The sepals are generally green. Then you have the petals. And then on the inside, this part is called the stamen. So one stamen includes the anther, which is the top part. That's the part that is going to produce the pollen. And then the filament, which is holding it up. And then the carpel, which is this part. This is the female part. It includes a stigma on the top, a style, which is the long, skinny part, and the ovary, which is the outside part of this, and then the ovule, which is inside. And there can be multiple ovules. My dog is going nuts. And if you have multiple carpels, in other words, more than one of these, like in this one, if they're fused, they are called a pistil, right? So the other flower is kind of a little more normal, but you can't tell the sepals and petals easily apart because they're not colored. And some flowers, they really are not. Like a lily, for example, you can't tell the difference between a sepal and a petal. So this has all of the features except for the receptacle, as in many flowers, has the ovary and the stamens and all other flower parts really above the receptacle. So they're coming out kind of at the top. This has just one carpel, so there's no pistil involved in this particular flower structure. All right, so the other thing that's going to happen is that ovary, whoops, I should have left that on there. So this ovary wall, what's going to happen is it's going to develop as the seed develops into a fruit. All right, so fruit and possibly other flower parts, and we'll talk about more about this in the lab, develop into fruits that are specialized for protection of the seeds and also for dispersal of the seeds. The ovule, which is inside an immature ovary, it will be fertilized and become the seed, and the wall of the ovary then becomes the pericarp. This can be either fleshy or it can be dry, and again, we'll talk more about fruits later. And if it's not fertilized, the whole flower will fall off. All right, so we're going to talk about the angiosperm life cycle. All right, so first of all, we'll just kind of generally go through, as in all the life cycles that we've talked about so far. A flowering plant, the main structure of the plant is sporophytic. So the whole plant, including the flower, gosh, my dog is nuts, is the sporophyte. And it is going to have the male part is going to be produced within the anthers. So here's the anthers, and the female part is within the ovary. All right. So the male part is going to, so this is like the microsporophyll that has the microsporocytes in it, like we drew out before, and it will undergo meiosis and produce 4 haploid cells, and those haploid cells will then divide via mitosis and make a generative cell and a tube cell. The generative cell then will divide twice and make 2 sperm cells. So this is similar to what we talked about with the gymnosperms. All right, so pollen for gymnosperms and angiosperms kind of develops the same. What's a big difference is how the ovule or the megasporangia or megasporocyte will develop. All right, so what's going to happen is the megaspore will undergo meiosis and make 4 haploid cells. All right, now those 4 haploid cells, one will survive and the other 3 do not, just like in the gymnosperms, so that's not different. But then what's going to happen is that one cell will do mitosis 3 times. So how many cells does that make? So it does it once, and that makes 2 cells. It does it again, that makes 4. It does it again, that makes 8 cells. 3 of them are going to migrate to the top. 3 of them are going to migrate to the bottom, and 2 of them to the center. All right? So this is in the megasporangia, right? Like this is that whole female gametophyte then. It is a multicellular gametophyte, just like the pollen grain is. And one of these down here, these 3 are called synergids. One of them is going to be the egg cell, and it's random which one. And the other 3 at the top are called antipodal, and they will just basically be broken down. And then the middle 2 are called polar nuclei. All right. So what will happen then is as the pollen grain grows, so this is the pollen tube, and it's going to distribute the 2 sperm cells through the micropyle, which is through this little part, into this section, one of which will fertilize the egg, and it becomes a zygote, and the other one fertilizes the 2 polar nuclei, and that becomes the endosperm. So what that makes the endosperm is 3N, and the zygote is 2N. All right, so that's kind of unusual. This is called double fertilization because there's 2 of the, both sperm are actually used, and they are doing different fertilization of different parts of the megasporocyte or megaspore. All right, so then that develops into the embryo. The embryo is part of a seed, and then that seed will drop on the ground and grow into the sporophyte. Now this particular drawing isn't showing this plant being a large plant, but generally speaking, it's a fairly large plant. All right. So one thing that I wanted to make sure that we go over here is that we look at this a little closely. The micropyle is between the integuments of the ovule. Now, I don't know if you can really see this very well, but look, there are 2 integuments. Remember way back I was talking about how the gymnosperms had 1 integument, and the angiosperms have 2. All right, so that's just a double layer of protection. It makes this little hole here through which the micropyle will grow. Here are the 2 sperm nuclei. One's going to fertilize the egg, and the other one fertilizes the polar nuclei, making a 3N structure, as we talked about. All right, and then lastly here, as this basically we get like this little zygote forming, this endosperm forms a nutritive structure around the zygote. So it's kind of like a placenta, so to speak. It isn't, but it's kind of like, in terms of the seed, it's very supportive of that zygote and helps keep it from drying out also. And then the zygote is going to kind of start growing. This is just showing you what the zygote does. It does mitosis, makes two cells, then it does mitosis again, then it does it again. So there's different parts of this, and if it's a eudicote, it's going to form two cotyledons, or two seed parts, and those seed parts are going to help give this plant some nutrients before it starts, as it, when it drops on the ground, before it starts doing photosynthesis. It's going to hold starches and so forth that are going to enable this plant to respire. And then once it starts doing photosynthesis, then it makes its own sugars and it respires it, and etc. All right. Okay. Let's go through the diversity of the angiosperms. There's what they call basal angiosperms. These are kind of they're not very advanced, so to speak. Some of them only have tracheids, not vessel members. Now, we haven't talked about that yet, but tracheids are really small vessels, and they occur in gymnosperms. Gymnosperms do not have larger vessels called vessel members, and all the other angiosperms do. And then water lilies, they don't have a lot of vessels either because they're in the water, so they don't really need that. And star anise doesn't have a whole lot either. It has some vessel members, but evidently not a lot. All right, and then the next group is magnoliids. These, of course, are magnolia, but also includes laurels and black pepper. And what's different about them, their flower, instead of the whorls coming off in one place along the branch, they would come off in a spiral. So that's kind of unusual and different. And the next two groups are monocotyledon and eudicotes. Now, both of these guys have true flowers, like we talked about. Their flower structure will be roughly the same. And there are a lot of representations. I would say like 80% at least, maybe more, are actually eudicotes in the environment naturally. And we have some monocots there, but because we plant a lot of monocots like grasses and palms and things like that, there's a lot more monocots. Things also like aloe and that sort of thing are also monocots. So we're going to compare monocot and dicot a lot as we go through the tissues and all of the plant structures, because they're really common plants that you would see out there. All right, so some basic differences. The pollen grains of the monocot only have one pore, whereas eudicots have 3 pores. Their leaves, monocot leaves, are parallel veins, whereas eudicots are netted venation. Their flower parts are in 3s, whereas eudicots are 4s and 5s. And other differences in their growth and their tissue arrangement will also be apparent as we go through the cross sections and talk about growth. All right. So one last thing is the importance of, let me just kind of put this up here. This is just in your book, and it's showing you maybe some pictures of these different guys. These are the basal angiosperms. And these are the magnoliids, and I think most of you probably know what a magnolia looks like. And I think you guys know what a palm and a grass looks like, and an oak tree, right? And then this is just a kind of telling you how many species of different things. You can see that mosses, there's quite a bit of them, as well as this is the fern group, the Monolithophyta. I can never say that right. All right, anyway, that's the old name. And there's a whole lot of ferns. Now, these guys evolved first, so that might explain it. But notice then flowering plants. Okay, so flowering plants have a whole lot of diversity. And there's just a ton of them. And in the ecosystem, there's many, many, many of them. All right? Now, in terms of importance, of course, all of our crops, not all of our crops, most of our crops are food. Our angiosperms. So most of our food is coming from angiosperm group, or at least flowering plant group. And I don't think there's that many. I don't think there's any food that's in the gymnosperms that I can think of. There's spices, and my dog is freaking out. Sorry. There's spices and wood, of course, that we build things out of and or make. things out of, like cabinets and so forth. And then, of course, there's medicines, and there's a table in your book talking about different medicines that are derived from seed plants. And you can see there's a whole lot of them. Much of the medicines are found actually as secondary metabolites that I mentioned earlier that are a lot of times found in the large central vacuole of a plant cell.